Welcome to our video, which will show you how to set up and carry out the GCSE Biology required practical investigating the effect of pH on enzymes. In this practical, we'll investigate the effect of amylase on starch at different pHs as indicated by the colour change of iodine solution. For this practical, you'll need either an electric water bath or a 400 millilitre beaker that you can turn into a water bath. You need a test tube rack and some test tubes, a range of different buffer solutions with different pHs, and also a thermometer. You'll also need some iodine solution, starch solution, amylase solution, safety goggles, some disposable pipettes, a glass stirring rod, something to mark your test tubes with, either a China graph pencil or a permanent marker, a spotting tile, some syringes, and a stopwatch. The first stage is to set up a water bath by heating some water in a 400 millilitre beaker until it reaches 35 degrees C. Alternatively, you could choose to use an electric water bath like the one I'm using here. We then label 5 test tubes with the pH value of each of the buffer solutions that we're going to use. We then add 2 millilitres of each buffer solution to the appropriately labelled test tube. So that's 1 millilitre of pH 7 solution and one more, so that we've added 2 millilitres to each tube. The 5 different buffer solutions are then transferred into the water bath, whether that's an electric water bath that you're using like me, or it could just be the beaker of water that you've heated up to 35 degrees C using the Bunsen burner. The next stage is to label 5 test tubes as starch, and your students will measure out 4 millilitres this time of starch solution into each of the 5 test tubes. They then need to transfer the starch solution into the same water bath alongside the different buffer solutions. Your students would then need to label 5 more test tubes with the word amylase, and this time we're transferring 12 millilitres of amylase into each of the test tubes, and then they also need to go in the same water bath along with the buffer solutions and the starch solutions. We then place a thermometer into one of the test tubes so we can monitor when the solutions have got to the desired 35 degrees C. Your students then add a few drops of iodine solution to each of the wells of the spotting tile. And in the first well, they also add a few drops of starch so that they've got a colour comparison to compare to for the rest of the practical. Once the solutions have got to the 35 degrees, we use a pipette to transfer 2 millilitres of the first buffer solution into one of the tubes of starch, and then we give it a stir with a stirring rod to ensure that it's thoroughly mixed. The students then use a pipette to transfer 2 millilitres of the amylase solution into the starch solution and start the stopwatch and stir it with a stirring rod. Once 10 seconds has elapsed, you use the stirring rod to transfer a drop of the solution onto the spotting tile and note the colour, making sure you rinse the stirring rod in between each 10 seconds. This is repeated every 10 seconds, remembering to rinse the stirring rod in water in between each time. This continues while ever the iodine changes colour, but then we stop once the iodine stops changing colour. The number of drops is recorded in a suitable table, and this process is then repeated for the other buffer solutions. Your students would then multiply the number of drops by 10 to get the number of seconds it took for the amylase to fully digest the starch. We hope you found our practical advice useful. For expert technical support, please contact the Philip Harris Technical Support Team on techsupport@philipharris.co.uk or visit our blog at philipharris.co.uk/blog.